Hi everyone, my name is Nikhil Kota, and I'm a sophomore biology major at Seton University. I'm also an undergraduate researcher at the Institute of Neuroimmune Pharmacology. The project I will be presenting today is binge exposure to ethanol modulation of stress genes in the hypothalamus of F344 rats. This project was done in conjunction with Dr. Hodgkinson, Dr. David Goldman, the clinical director of the National Institute of Alcoholism and Alcohol Abuse, Dr. Liu, Wenfei Huang, and Dr. Suli L. Cheng, the president and director of the Institute of Neuroimmune Pharmacology, as well as my mentor. In the United States, 25% of people ages 12 years or older have reported binge drinking, which is drinking to a blood alcohol concentration level of higher than 0.08 milligram per ml. At the same time, alcohol is known to cause spleen atrophy, which is shrinking of the spleen, which is an essential immune organ that filters red and white blood cells. In outer space, astronauts feel a phenomenon known as microgravity, in which they feel weightless. Previous studies through NASA have determined that this phenomenon actually reduces their immunity. Therefore, any potential astronauts with a history of binge drinking or prolonged use of alcohol have a higher risk of contracting an immune-related infection in outer space, since the immune system is already compromised in outer space. So the purpose of this experiment is to determine how binge exposure to ethanol induces spleen atrophy, and if and to what extent it increases the expression of stress-related genes. As similar to alcohol, stress is also a confounding variable for immune-related infections. So this experiment used 3 different age groups of Fisher 344 rats. The F344 rat model is a strain of rats that was developed in the 1920s for immunology and cancer research. The 3 different age groups were an adolescent age group, a young age group, and an adult age group. The adolescent age group had a postnatal day, or PND, of 34 to 39. The male young rats had a PND of 60 to 65, and the male adult rats had a PND of 86 to 91. So these rats followed a 6-day schedule in which the first 2 days they were given intragastric or IG injections with water for conditioning. On days 3 to 5, they received 52% weight by volume ethanol, and on the final day, day 6, they were sacrificed and their spleens and brains and thymus were collected. Following a single spleen cell isolation, flow cytometry was used to immunophenotype the spleen cells. So in other words, determine how many T cells or B cells the spleens had. Following the immunophenotyping, the hypothalamus was collected from the brain. Its RNA was extracted, visualized via gel electrophoresis, and was converted to cDNA via reverse transcriptase PCR. Although this study we were not able to fully research the stress gene response, future studies will. But for now, we are just going to focus on the developmental data. So when we look at the percent survival of the rats at different ages, you may notice that the male adolescent rats had the greatest decline with about 70% survival, whereas the older male rats had 100% survival rate. And if we look at the relative spleen weight, which is spleen weight over body weight, between the male and female adolescent rats, you may also notice that the male adolescent rats have a significantly more decline than the females. At the same time, the female adolescent rats actually have a similar response, similar to the young male and male adult rats. We hypothesize that the reason why the female adolescent rats had a less severe effect is that since they experienced puberty much earlier in the adolescent stage than the male rats, they were less prone to the atrophic effects of binge ethanol. Following the immunophenotyping assessment, we were able to see that there was a strong correlation between the percentage of natural killer cells and relative spleen weight, especially in the adolescent males. The adolescent males had an R-squared value of 0.899. The young male rats had an R-squared value of 0.68, and the adult males had relatively low correlation value of 0.075. So we saw that the correlation between the percentage of natural killer cells actually decreased as the male rats aged. So from this study, we have three main conclusions. One, the male adolescent F3-4 rats experienced the greatest spleen atrophy. Two, the natural killer cells could be the underlying cellular mechanism for spleen atrophy. And three, spleen atrophy is actually puberty dependent, as since female adolescent rats are developmentally more mature, they experience less severe spleen atrophy. Future works should identify the cellular pathways at which binge ethanol impacts natural killer cells. Future studies could also research the puberty-based differences between the male and female adolescent rats. And finally, although due to a time constraint we were not able to include the stress-related gene data, that will definitely be a future study. Thank you all for listening to my presentation. Have a good day.